mitochondria at the center of all of this is probably, in my view, the most relevant biochemistry that exists in our area right now. And we need to understand a lot more about it if we actually want to solve the problem of this multitude of metabolic diseases. Now, as we start our discussion about this central biochemistry, which is fructose uric acid metabolism, I want to preface the entire discussion with the idea that this is an evolutionarily conserved biochemical pathway, which is integrated with glycolysis and its own connections to the mitochondria. And why this was important to humans from an evolutionarily important aspect is because when we take in certain macronutrients, we're going to talk about that in elevated quantities, we actually have a shift from one type of metabolism that in this discussion I'm going to call burning to another type of metabolism, which I'm going to call storage, that is complete with systemic inflammation, elevated fat synthesis along with glycogen and the storage of water, and the alteration of nitric oxide signaling, which alters our blood pressure. Dr. Richard Johnson has talked and written about this extensively, and I provide you there with two references. When we activate this metabolism, which from the point of view of evolution was important to our survival because it allowed humans to the capability to rapidly put on fat, store glycogen and water, it drove our foraging capabilities, hunger, thirst. It elevates insulin resistance because that is necessary to make sure that the nutrient load coming in gets to all the relevant tissues that need it. We have the elevation of the systemic inflammation because we want to activate or stimulate the immune system. And then we have the elevation in blood pressure in order to make sure that all of the nutrients get where they need to go. And this is a natural aspect of being human. The problem with today in 2023 is that this metabolic pathway, this what I'm going to call the red state, is being activated chronically, day in, day out, week after week, month after month, year after year. And this is how we get to the constellation of metabolic diseases, which I'm going to turn to now. We know when this core biochemistry is activated, that fructose uric acid metabolism is the driver of not only gout, but also kidney disease, diabetes, obesity, fatty liver disease, Alzheimer's and dementia, cardiovascular disease, coronary artery disease, and also sugar and alcohol addiction. And we know this from the body of Johnson's work, which is over 700 publications based on cellular and molecular data, based on animal studies of a wide variety, and also clinical studies in humans. On this idealized slide, glycolysis is depicted in gray on the left part of the slide. The polyol pathway with its connection to fructose uric acid metabolism is shown on the right side of the slide in red. Our genetics predict that the polyol pathway with its connection to fructose uric acid metabolism is potentially available in all of our tissues and organs. Therefore, the functionality of these two pathways will depend on the nuanced function of a particular type of tissue. The main function of glycolysis under low glucose conditions, these would be conditions that are 5 millimolar intracellular glucose or less, which is about 90 mg per deciliter of glucose, is to produce substrate level ATP required for basic cellular function and the mitochondrial substrates, pyruvate and lactate, which will then enter the mitochondria and be burned to produce ATP. Lactate is in fact the terminal product of glycolysis and is formed in a ratio of about 7:1 lactate to pyruvate. Under these low glucose conditions, fatty acids that are in the cytoplasm can enter into the mitochondria where they are burned by beta oxidation to produce ATP. This is a cellular state that I characterize as burning, and I will refer to it in this video as the white state. Now, as glucose rises, as it would during a standard American meal, and by the way, we are going to focus on the liver as our model system for this part of the discussion. And as the glucose rises, polyol pathway opens, and we start to push glucose into endogenous fructose production and open fructose uric acid metabolism. As the fructose is formed, it is immediately phosphorylated, and fructose uric acid metabolism lacks all of the speed bumps that you normally find in glycolysis. There are no feedback mechanisms that slow the progression. So fructose uric acid metabolism opens. We have the sudden decline in cellular ATP along with phosphate. We have a sudden acute rise in uric acid, which is a signaling molecule. We activate de novo lipogenesis, fat synthesis, the production of glycogen, along with the storage of water. We have an elevation of systemic inflammation, activation of JNK1 along with IL-1 beta, and we have dysregulation of the nitric oxide pathway at the level of the liver, which downstream has its effects on the cardiovascular system, one of which is the elevation of blood pressure. Additional activators of fructose uric acid metabolism, aside from the glucose issue and the polyol pathway, include exogenous fructose coming in with added sugar, and added sugars can include both glucose and fructose. We also have alcohol as a major or profound activator of fructose uric acid metabolism, in addition to processed food and processed meat, which usually include glutamate, inosine monophosphate, adenosine monophosphate, in addition to salt and added sugar. And there is evidence to show the expression of these pathways in brain, adipose, muscle, intestine, liver, endothelium, the chondrocyte, which is the biologically important cell that works in our joints, heart, and pancreas. This slide demonstrates the type of literature that you now find in regard to the polyol pathway as a glucose sensor and indicator of a metabolic switch, a metabolic shift from burning to storage. Polyol pathway signals a shift from the white state to the red state, where the red state is characterized by fat storage, glycogen storage, the storage of water, systemic inflammation, and the alteration of nitric oxide regulation. I have made the assertion that under conditions of low glucose, under 5 millimolar glucose, which is approximately 90 mg per deciliter, that the white state dominates the situation. This is the state of burning. But when we have a meal, a standard American meal that is high in glucose, and we shift the intracellular glucose above 5 millimolar or above 90 mg per deciliter, that we open the polyol pathway, which funnels into and opens fructose uric acid metabolism. I've made that assertion.